Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning session in plant cell and tissue culture. Today in this session we learn about protoplast isolation. In this picture you can see the images of plant cell under microscope. So this is the picture of plant cell under microscope having the protoplast covered with thick cell wall made of cellulose. And from this plant cell, when we isolate the protoplast will look like this. It will be very fragile, circular in shape, and will lack the cell wall. So these protoplasts are the cells without the cell wall. They are the naked cell without the cell wall. So these protoplasts will be isolated from any type of the tissue in order to fuse the two protoplasts having the desired characteristics. So this protoplast can be isolated by two important methods. First method is mechanical method. The another method is enzymatic method. First, we'll take up the mechanical method. You take the leaf tissue which you want to isolate protoplast from that tissue, and then you incubate in the plasmolysing solution. And after incubating in the plasmolysing solution, cut the tissue with the sterile knife and you isolate the protoplast. And here the yield of the protoplast obtained will be very poor, and most of the time it will be ruptured. And these ruptured protoplast will affect the intactness of other protoplast. And this technique was first reported in 1892 by Klecker. So this is mechanical method. The another method is enzymatic method. So this method was demonstrated in 1960 by the scientist Cocking, and this technique was used to isolate the protoplast from the higher plants. Using the enzyme cellulase, which was isolated from the fungus Myrothecium verrucaria. So here the tissue will be taken and it will be incubated with the enzymes which are isolated from the fungus, bacteria or other source. Here the yield was more compared to mechanical method. But this mechanical method was applicable only to vacuolated cells to isolate the protoplast. But this enzymatic method could be applied for any type of the higher plant protoplast isolation. However, the real progress in the area of enzymatic isolation of the protoplast was made after 1968, when cellulase and macerozyme enzymes were available commercially in the market. Later, in 1968, Takebi et al. Were the first group to use these commercial preparations of the enzyme to isolate the protoplasts from the mesophyll cells of the tobacco. Here, this enzymatic method, the tissue will be taken and will be incubated with these enzymes, which are isolated from the different sources. After that, they will isolate the protoplast. So this enzymatic method of isolation of the protoplast are of two types. One is the one-step method and another one is sequential enzymatic treatment for the isolation of the protoplast. In one-step method, what they do is the tissue will be taken and the epidermis will be removed and it will be incubated in the plasmolysing solution, and to this plasmolysing solution they will be adding all the enzymes like pectinases, cellulases, etc. So all the enzymes will be added. Pectinase will act on the pectin, cellulose will be digested by the cellulase, and the protoplast will be released. And later this protoplast will be purified and cultured on the medium. So this is one step method. In sequential enzymatic treatment, this plasmolysed leaf tissue will be first treated with the pectinase, and pectin will be digested, and these released cells, whatever the pectin digested, the suspension is there, to that cellulase will be added, remaining enzyme will be added, and like this the sequentially the tissue will be degraded and the protoplast will be isolated. However, this one step method is now widely used to isolate the plant protoplast. It's a routine process, that is the one step process wherein you take the tissue, plasmolyze it, add all the enzyme and incubate, and you take the protoplast purified for culturing. With the available enzymes in the market and the techniques available, viable protoplasts can be isolated from any plant organ or the cultured cells, provided they have not acquired lignification. Protoplast isolation has been reported from mesophyll cells of in vivo as well as in vitro grown plants, aseptic seedlings, embryogenic and non-embryogenic suspension cultures, cotyledons, hypocotyls, and male and female gametes. But this protoplast isolation is affected by many factors. This includes source of material, enzyme treatment, osmoticum, all these play an important role in getting the good yield of the protoplast. First, we'll take up the source of material. The most widely used tissue to isolate protoplasts are the young leaves from the in vitro grown aseptic shoot cultures. Since these mesophyll cells in the leaves are loosely arranged, the enzymes have an easy access to the cell wall. Moreover, the leaves yield large number of uniform cells, whereas leaves do not yield culturable protoplast as in case of cereals. Their actively growing cell suspension cultures are used. To obtain totipotent protoplasts of citrus, usually embryogenic suspension cultures are used. So like this, source of the material will be varying from one type of the organism or one type of the plant to another. But if you take the softer tissue and having the cells which can be easily digested by the enzyme will be useful for isolation of the protoplast. So this source of material affect the isolation of the protoplast. The second factor is enzyme treatment. The isolation of the protoplast requires at least two enzymes like pectinase enzyme to dissolve the middle lamella that binds the adjacent cells together, and second enzyme is cellulase enzyme to digest the cell walls and release the protoplast. Some tissues may also require hemicellulase in addition to cellulase and pectinase to release the protoplast. Several brands of cellulase and pectinase enzyme of the bacterial origin and fungal origin are commercially available. And these are isolated from the bacteria like Trichoderma viridae and fungus like Rhizopus and Aspergillus niger. So few commercially available enzyme mixtures are like cellulase R10. So they will be giving the commercial name depending on the company. The the commercially available enzyme contains various impurities, including proteolytic enzymes, phenolics, and salts, which may adversely affect the yield and viability of the protoplast. Therefore, some workers have recommended the purification of enzyme by eluting through biogel Cephedex method or treatment with activated charcoal. However, the most of the enzymes have been used in their crude forms as available in the market with satisfactory results. So like this, the impurities which are present in the commercially available enzymes, they affect the yield of the protoplast. Next, another important thing is osmoticum. Osmoticum is the chemical like mannitol, sorbitol, sucrose in combination with the glucose. So these will be used in around 0.7 to 0.8 molar, and they maintain the osmotic strength of the solution in order to maintain the viability of the protoplast. We all know, unlike cells, the isolated protoplasts are osmotically fragile. Therefore, right from the enzyme treatment to the culture of the protoplast until they develop a substantial cell wall, proper osmolarity is maintained around them. In a solution of suitable osmolarity, usually hypertonic solution will be maintained. The protoplast appear completely spherical. A variety of solutes, ionic and non-ionic osmotic, have been used for isolation and culture of protoplast. However, the most commonly used osmotic stabilizer or osmoticum is the mannitol, metabolically inert sugar, and it is used in concentration of around 800 millimolar or zero 0.8 molar. So this osmoticum play an important role in maintaining the viability of the protoplast. So all these factors play an important role in protoplast isolation. So after isolating the protoplast by enzymatic method, you cannot use the same suspension directly on the culture media. You have to purify the protoplast. So this protoplast suspension, what you have got after enzyme treatment, will be having the undigested material, that is the debris. It will be having the burst or the opened protoplasts, and it will have the enzyme which we used for the isolation of the protoplast. Here what they do is, initially the suspension will be, it will be passed through the metal or the nylon sieve. It will be passed through the metal or the nylon sieve to remove the large debris. So it will be filtered through the metal or the nylon mesh to remove the debris. Next, enzymes are removed by centrifugation and protoplasts settle at the bottom of the tube, and the supernatant is removed with the help of the pipette. So whatever the pellet you have, that will be the protoplast. And intact protoplasts are separated from the broken protoplasts through the centrifugation, and again the suspension will be removed by the pipette. This is how the isolated protoplast will be purified. So after purifying the protoplast, we have to check the viability, whether it will multiply or grow on the in vitro media. So for checking the viability, just like our suspension cell culture viability, how we check, here also the same techniques will be used. There are many methods to check the viability of the protoplast. The first one is phase contrast microscopy. Here, microscopic assessment of the cell viability is based on the cytoplasmic streaming and the presence of a healthy nucleus. while the phase contrast microscopy gives a better picture of these features. It is often not difficult to observe them under bright field microscopy. Then another method is tetrazolium salt or the tetrazolium test. In this test, the respiratory efficiency of the cells is measured by reduction of TTC, that is 2,3,5 triphenyl tetrazolium chloride to red dye, that is formazan, which can be extracted and measured spectrophotometrically. They will add the TTC to the isolated and purified protoplast. If it turns red color, that is due to the formation of the dye formazan, so that will indicate the presence of viable protoplast, so that can be measured spectrophotometrically. Although this method allows quantification of observation, it alone may not always give a reliable picture of the cell viability. For that, they go for the FDA test. FDA means fluorescein diacetate test. This technique offers a quick visual assessment of the percentage of the viable cells or the viable protoplast. Here, the stock solution of FDA, fluorescein diacetate, at a concentration of 0.5% is prepared in acetone and stored at 0 degrees centigrade. To test the viability of the protoplast, the solution is added to the cell or the protoplast suspension along with the osmoticum at a final concentration of 0.01%. After 5 minutes of incubation, the cells are examined under fluorescent microscope. FDA is non-fluorescent and is non-polar, and it freely permeates through the plasma membrane. Inside the living cell, it is cleaved by the activity of the esterase enzyme if the protoplast is viable and releases the fluorescent polar portion, fluorescein, which fluoresces in green color. So that will be the indication that the protoplast is viable. So if the viable protoplast turns the FDA solution into green, then it indicates that the protoplasts are viable. That is how the viability of the protoplast will be checked. After checking the viability, that will be culture on the aseptic medium. The basic principles of protoplast culture is the aseptic isolation of large number of viable and intact protoplast, moving their cell wall and culture them on the suitable nutrient medium. Here you can see the picture where the leaf tissue was taken and it was peeled and plasmolized, and from that there is isolation of the protoplast pellet, and here they've shown the fusion of the protoplast that you will be learning in the coming session, that is protoplast fusion. For that I will make the different video that you will be learning it. So the isolated protoplast will be inoculated on the media, and after you plate it, it develops the cell wall and it starts dividing, and it will form the clump of cells, and colony will be formed, and it will form the undifferentiated mass of cell that is called callus. From that, callus differentiation takes place and there is formation of the complete plantlet. Later, this the regenerated plant under the in vitro condition will be transferred to the greenhouse condition. Here, the viable protoplast regenerate a new cell wall within 48 to 96 hours after isolation. The protoplast that fail to regenerate wall generally will not divide and it will die eventually. Also, all the healthy protoplast may not divide, and therefore plating efficiency is calculated to estimate the cell vigor. The what is this plating efficiency is the number of dividing protoplast per total number of protoplasts plated. How many are dividing out of the total plated or inoculated protoplast? The protoplasts capable of dividing undergo first division within 2 to 7 days after isolation. The delicate nature of the protoplasts demand modification of the Murashige and Scoob media or the B5 media. Besides higher osmotica, the inorganic salt concentration is adjusted. Inorganic salts like ammonium nitrate concentration is lowered and calcium level is increased. And more of organic components, vitamins and plant growth promoting compounds are added in order to hasten and promote the cell wall synthesis. Due to the sensitivity to the light, protoplasts are cultured in diffuse sunlight for initial 4 to 7 days. After early culture, when protoplasts have regenerated new cell wall and divided, they are transferred back to normal medium where plants regenerate by shoot formation or somatic embryogenesis or by the callus formation. This is how protoplasts are cultured. So the isolated protoplasts can be cultured in several ways, of which agar embedding technique in small petri dish is commonly followed. Agar embedding technique will be done to culture the protoplasts. In this technique, protoplast suspension is mixed with equal volume of the melted agarified medium, and the protoplast agar mixture is poured onto the small petri dish. In petri dish, embedding of protoplast in solid agar medium is known as plating of the protoplast. In culturing the protoplast, so when we are plating and growing, there is a reformation of the new cell wall around them. Then the cell form the cell with the intact cell wall will be formed. The protoplast subsequently enters the cell division. Cell division takes place only after the development of the cell wall, which is followed by the formation of the undifferentiated mass of cell, that is callus. So such callus retain the morphogenesis and plant regeneration, and this callus will be developing into new plantlet. This is how the protoplast will be cultured on the medium. So in this session, we learned how the protoplasts are isolated, the methods of isolation of the protoplast, the factors affecting protoplast isolation, and we concentrated on how the viability of the protoplast will be checked and how it will be grown and will be cultured. I hope you understood this session. Thank you.